In interphase 1, the chromosomes replicate. The two strands in the nucleus represent the two strands of DNA from the mother and the father. The chromosomes are not visible under a microscope. The green and yellow colors represent the chromosomes. The orange represents the nucleus, and the blue dots represent the centricles.
In prophase 1, the green and yellow chromosomes condense and eventually attach to their counterpart. Also, each one of the chromosomes make an X-like shape. This permits the DNA to share and give genes to one another. Also, around the centricles, spindle fibers begin to form around it, which then the centricles begin to move to opposite sides.
Despite us showing only 4 chromosomes in the cell, 2 pairs, there are 23 in reality. In prometaphase 1, the fibers from the centricles link on to one of the chromosomes. Also, the chromosomes are moving while the centricles move to opposite poles. The nucleus also dissolves.
In metaphase 1, the centricles eventually meet their destination and arrive at opposite sides. This causes the cell to stretch out and pinch in the middle. Also, a metaphase plate is formed, and the chromosomes line up on either side of the plate, not on the plate.
In anaphase 1, the indent in the cell continues, causing the cell to stretch more. The chromosomes on each side start moving toward the opposite ends and they eventually split. However, the chromatids do not split.
In telophase 1, the chromosomes get to opposite ends and go to the newly formed nuclear membrane. The centricles are also in the nuclear membrane. The cell continues to prepare to divide.
In cytokinesis, the cell divides into 2. This is eventually basically when the cell divides. The centricles also move.
In interphase 2, there is no DNA replication. The DNA is not replicated because it was already replicated in interphase 1. In prophase 2, the chromosomes condense and the 2 pairs of centricles separate into 2 chromosomes. The daughter centricle also attaches to each of the original centricles. The chromosomes do not attach to the nuclear membrane, which permits the exchange of genetic info.
In prophase 2, there still are 2 cells, but we only are looking into one of the cells to make it easier. Here the spindle fibers attach to the chromosomes. The chromosomes are attached to the fibers. The centricles have now finished migrating to opposite poles and opposite sides. In metaphase 2, the centricles pull onto the chromosomes from both sides. This creates opposite attraction, so basically the chromosomes are being pulled from different forces. The chromosomes also form in a line along the metaphase plate due to the forces being pulled onto them.
In anaphase 2, because the centricles are pulling from both sides, the chromatids go apart and move towards opposite sides of the cell.
In telophase 2, the chromatids reach opposite sides and lie on the nuclear membrane. Go.
In cytokinesis, the cell divides into 2 cells, 4 in reality. Because of this division, there are now 4 daughter cells, and each cell has a different set of chromosomes.
In reality, there are 4 different cells. However, we chose to use one because we thought that it would make it easier to understand and follow.